Hello, my name is Kip Balkcom. I'm a research agronomist with USDA ARS located in Auburn at the National Soil Dynamics Lab. I'd like to talk to you about a study we have that's been ongoing where we are examining cover crop mixtures preceding corn. Specifically, we want to take a look at the biomass production associated with these different mixtures and single species cover crops, as well as looking at how they affect early season corn growth and yield. In addition, because these mixtures contain legumes, we want to determine if we can reduce the nitrogen requirements for the corn following these mixtures. Now, just to give you an idea about the treatments that we had in this particular study, it's pretty simple. We have 5 different cover crops where we're looking at fallow with no cover, where it's weed free. We sprayed it to make sure there were no weeds there. We have rye, which is kind of our control cover crop. We did drill that. All the covers are going to be drilled at 90 pounds to the acre, and we did fertilize the rye with 30 pounds of nitrogen to the acre, and this usually occurred the 1st of February to make sure that we had adequate biomass production. Then we had a pure crimson clover stand at 20 pounds to the acre, and then we also compared a mixture of rye at 30 pounds to the acre plus clover at 10 pounds to the acre, as well as a second mixture that included rye at 30 pounds to the acre and clover at 20 pounds to the acre. And the way that you can remember this, I'm going to refer to this throughout the presentation. Mix 1 would be 10 pounds, mix 2 would be 20 pounds. And then for each one of the cover crops, we actually applied different nitrogen rates across there. We had nitrogen rates ranging from 0, 150, 200, and 250 pounds of N to the acre. Now, as I mentioned, this is kind of an ongoing study. We've had this at several locations. I've got 6 site years here that I'm going to summarize. You can see primarily a lot of these have been at E.V. Smith, but we have had a couple of sites here at Headland where I'm at today. And I want to talk about the plant dates and the termination dates associated with the cover crops. You can see in general, we had these cover crops planted by the middle of November every year. And then we had our termination dates corresponded to around the 3rd week in March every year. And then that allowed us to have a corn planting date approximately the 1st week of April. Now, that's probably a little bit later than what a lot of people would plant their corn, but we were trying to maximize the growth period for these cover crops as much as possible. That's one of the problems that we have with corn or any kind of cover crop preceding corn. It's always the first crop we plant in the spring, and so we have a very compressed time frame where we can actually plant a cover crop and get decent growth out of it. Now, I want to summarize the biomass of the cover crop mixtures, and I'm going to set this graph up. It's going to be the way that it is for all the other graphs following it. It's called a heat map. It's pretty easy to just present a lot of information quickly. On the traditional y-axis, we have the environments of the site years, which would correspond to, say, E.V. Smith 2016. And then we also, on the x-axis, we have the different cover crops. And then each block with a number corresponds to the biomass that was measured, the average biomass that was measured for each location and cover crop combination. And then we have a color scale that helps us distinguish between high values compared to lower values. So the first thing that you can see on this particular graph is we have 2 locations, E.V. Smith 16 and Wiregrass 17, that had much lower biomass compared to the others. And I think that was just a function of the climate that we experienced at those particular locations. The other thing that you can see is there's a lot of variability within these cover crops and within the years. We had significant differences across all the rows within a location, but it wasn't always the same cover crop was superior to the other. So another thing about this graph is you can compare and see how a particular cover crop performed across all the different locations. For example, mixture 2 had some of the highest biomass that was measured at a couple of locations compared to the others. Now, if we take a look, when we measured that biomass, we also measured the nitrogen concentration that was present in the cover crop biomass. And so we can take the amount of biomass that was produced, multiply that by that concentration, and get a total nitrogen content. And I'm calling that uptake here, but you can see the uptake of these cover crops across all the different environments. And of course, again, the same 2 locations show up where we have lower N contents overall. And then you can see how the rye sort of separates out from the other mixtures because rye is not going to generally have a high N content compared to something that's a legume that's obviously going to have a much higher N content. The other thing to notice is there's a lot of variability. For example, crimson clover had nearly 100 pounds of nitrogen in that residue at one location, but then it dropped to as low as nearly 10. So there's quite a bit of variability in the N contents of this residue. Now, the thing about it is, this residue that's available in this cover crop doesn't mean it's going to all be available for the subsequent crop. It's not going to be 100% efficient. You have, just like when we apply fertilizer, you generally have losses that may occur and so forth, and it's never 100% efficient. That's the same way what we're dealing with the cover crops as they break down. Now, of course, one of the things that controls how quickly these residues release their nitrogen is something that I think people are probably very familiar with. It's CDN ratio, or carbon and nitrogen ratio. And this is just an example graph. This was just for one of the locations, E.V. Smith 17. And you can see here I have a blue bar that represents a range of 20 to 30 to 1. Everything below 20 to 1, that residue is going to decompose very quickly, and that nitrogen is going to be released pretty quickly and be available for plant uptake. Unfortunately, it could be available for environmental losses such as leaching as well. Now, you can contrast that with something that's over 30 to 1, which we would consider a higher CDN ratio. That residue is not going to break down near as quickly. The nitrogen that's present in the material is more than likely going to be consumed during that decomposition process, and it's not going to be available. So this just kind of follows what we would expect. We have a grass here that doesn't have a high N content, so it has a higher CDN ratio compared to a pure legume that has a much lower CDN ratio. So now let's take a look at what happens with the actual corn itself following these different cover crops. We looked at early season corn biomass, and in this instance, when I'm talking about early season, I'm talking about we came in about V6 around side dress time and measured the biomass of the corn plants across all these different plots. And so you can see here really not a lot of difference. The biggest thing that shows up is you did have one particular location that had much higher values than the other environments. And I think this was just a function of probably more than likely excellent growing conditions at that particular location that resulted in really good early season growth. Now, although these are all significant differences at a location, they're relatively fairly consistent. There's not a dramatic increase one way or the other for one particular cover crop versus the other. So if we take a look at that same uptake values for this early season corn biomass, you can see here the one thing that jumps out is notice the scale. We're at 25. We're much lower. So if you recall, we had crimson clover that had an N content of about 100 pounds at one of the locations. Well, you can see that did not translate into what the plants actually were able to take up. So it either was released and we lost it for whatever reason, or it didn't get released. But for whatever purpose, that's what I was talking about. It's not going to be 100% efficient. In this case, it was much below 100%. We didn't take up as much nitrogen as we would like to see. Now, if we carry this on over all the way to the corn yields, we just had two locations that were actually different from each other, but they kind of bounced around as far as what was the best treatment compared to the across the different cover crops. I think the biggest thing this shows is you see that E.V. Smith corn yields were maybe a little bit higher than wiregrass corn yields, but there's not a tremendous amount of consistent difference between the corn yields following these different cover crops. Now, overall, these yields I'd say are much lower than probably what people would expect, but I think it has to do with the way we conducted this test. If you recall, we didn't put any nitrogen out on this corn until side dress. We were trying to rely solely on the effect of the cover crops to supply nitrogen to the corn, and as a result, we more than likely lowered the yield potential because we put that corn in under some stress, just trying to get the effect of the cover crops themselves. When we take a look and average all the cover crops over the different N rates that were applied, the first thing that jumps out to me is there really was no response over 150 pounds of N to the acre. There was some differences between the environments. We had a little bit higher yield potential between some of the environments compared to the others, but overall we didn't see any kind of response above 150 pounds of N to the acre for the yields that we experienced. Now I did want to show this. This is a little bit busier graph. We're looking at an interaction between the cover and N rate, and the main reason I wanted to show this, we had a couple of locations where we did see a significant interaction, and notice the orange line here where we had 0 nitrogen applied. We were able to produce approximately 100 pounds of 100 bushels per acre of corn with no nitrogen, only the crimson clover. But unfortunately, that did not occur at all the locations. We didn't see that all the time. So it's very difficult to try to make a recommendation on something when it only occurred in one particular environment. It shows it is possible, but it just was not a consistent response across the environments that we had. So just to quickly summarize, we have our biomass production was very variable across these environments, and it was really inconsistent, meaning one cover crop might be good one year and then not as good the next year. The corn growth and yield following these mixtures and single species was very limited compared to the fallow. Sometimes it was better and then sometimes it was not. We just never saw a consistent response. The other thing is no appreciable amounts of N were supplied by the cover crops to consistently reduce N requirements across the environments we examined. Now we obviously set up this study to look at nitrogen effects from the cover crop, and we didn't see a lot of benefits from that. And primarily I think it's because of that short growing season that we have for our corn, for our cover crops preceding corn. There's just not a lot of time for it to produce biomass and actually produce a high N content that could be utilized for the subsequent crop. And that's not to say that a cover crop's not beneficial, because it could have other effects such as moisture control or erosion control and things like that. Obviously they're not going to be as great because we don't have as much biomass, but we were specifically looking at the nitrogen aspect associated with these covers. So I hope that summarizes this test for you, and I certainly appreciate your time today.